Ellis graduated from McMaster University in Hamilton for his bachelor, and then he did his PhD in chemical, bachelor and also PhD in chemical engineering at Northwestern University, Illinois. Then he did his postdoc in biophysics in University of Western Ontario, where he is a professor currently in the Department of Biophysics in the School of Medicine.
That's good enough.
Right. He's a recipient of Career Investigator Award from the Heart and Stroke Foundation in Ontario. He has also served as a graduate chair in medical biophysics, as well as being an associate dean for the School of Medicine and Dentistry. And yes, and he has had many journal publications, over 80 journal publications with highly cited. So please join me to welcome Dr. Ellis.
Thank you very much for inviting me, and thank you for giving me the opportunity both to be an examiner and of a PhD thesis that was successful, and to give a talk about my research. It's quite different. I doubt if there's anybody here work on the microvasculature? Anybody work on oxygen transport in the microvasculature? So this is so part of this lecture is going to be attempting to give you an education as well as talking about the research that I do. And I sort of put out a little bit of a title to pique people's interest maybe. Microvascular regulation of oxygen supply, the most important control system on planet Earth. Well, I don't know hardly anything about control systems. I'm sure there's people in the room who know a whole lot more about control systems than me. But maybe by the end of this lecture, you might agree with me that this one might be just the most important. We'll see. So I think you all know that oxygen is incredibly important in the body. If we don't have oxygen, we don't survive. If we don't have a cardiovascular system to distribute that oxygen around the body, then we're only going to be a few, maybe a millimeter or so in size, really small. So we can't have complex organisms without lungs, heart, cardiovascular system, and we also have to have a microvasculature because we've got to get the oxygen to exactly where it's needed. And we need a control system to be able to do that. So that's sort of an overall summary of what this is about. I don't do this work all on my own. I got a lot of people that I work with. Some of them are intensive care physicians because I also study various diseases. Some of them are engineers. Some of them are my research tech has been with me for so many years. We don't talk about it anymore, and she's indispensable to all the work we do. We're funded currently by CHR and NSERC. In the past, I've had heart and stroke and NIH funding. And some of the work I'll be talking about was with colleagues at St. Louis School of Medicine, Mary Ellsworth, Randy Sprague, and Alice Stevenson. And the modeling work, I think I showed a bit of the modeling, is Dan Goldman and Graham Fraser. So if you were to look at the microvasculature in skeletal muscle, so skeletal muscle in the hind limb of a rat, a light microscope, using the appropriate wavelength of light so we get high contrast, and magnification, this is a 10-power image, so that bar is about 100 microns. The muscle fibers are running in this direction, and we are looking at those little dark spots that you see moving through our individual red blood cells traveling through capillaries. So this is a real-time image in a living rat where we have cut the tendon and reflected. We've cut through the skin, cut the tendon on the muscle, reflected out onto the stage of the microscope, so we can shine a very intense light through it, and then look at it with an inverted microscope so we have a relatively high-power system to look at it. Digital video cameras give us nice, clean images and nice resolution. They've got amazing dynamic range for collecting this type of information. So there's our red cells flowing through capillaries. We picked the wavelength because red cells actually absorb a lot of light at that wavelength. This is where the blood flow is coming into this particular area. So there's a little tiny artery here called an arteriole, it's not really any bigger than a capillary, but it's supplying red cells to many capillaries. Then we've got the capillary network itself, and then up here at the top, we've got a venule where a venule is a very tiny vein that returns the blood back to the lungs for reoxygenation. Here it is at the higher magnification. We look at 20 power. You can see now quite nicely the discrete individual red cells in the arterioles that are supplying it. And then I'll just show you another. This is at the venular end where it's collecting, so it's collecting into a small venule and taking it away. Well, there's a man by the name of August Krogh back in the early 1900s, about 1915 or so. He's a physiologist in Copenhagen, and he was doing a lot of research on oxygen delivery to tissue. He first looked at oxygen uptake in the lungs, and then he started thinking about the periphery and thinking about if most people thought that delivery of oxygen in some way was an active process. They didn't really understand the principles behind it. He took a look at this problem and worked with a mathematician by the name of Erlang. They said, well, each of these capillaries is going to be a source of oxygen, and it's governed by a fixed law. It's going to be simple diffusion from those red cells out into the surrounding tissue. And as he thought about it, he realized, well, there's a limit as to how far that oxygen can diffuse. The tissue is consuming oxygen, and if I've got a particular oxygen level at this level, and the tissue is consuming it, the PO2s will fall off with distance away from those vessels. If I've got very low oxygen consumption, it'll extend very long ways that it can supply the oxygen. As the consumption increases, it will get closer and closer. It won't be able to supply oxygen into the tissue as far. And so we realized that supply of oxygen into the tissue is limited by simple biophysics, simple diffusion and solubility of oxygen in the tissue itself, and the rate of which it's being consumed by the tissue, and the diameter of the capillary itself. He then, as an engineer, how would you simplify this problem? You didn't have massive computer systems at that time that didn't exist. So they just thought about it in terms of simple diffusion in a one-dimensional diffusion from the vessel out of the surrounding tissue. And they defined a cylinder of tissue around that capillary. So my head's the capillary. They put a cylinder of tissue around it and said, how large a cylinder of tissue can that capillary supply? And depending on the consumption rate, that would be either large or really quite small. And he realized that as you increase consumption, that cylinder had to get smaller and smaller. So he said there's going to be active control of flow into each individual capillary. We have to be able to supply oxygen within 50 to 100 microns of where the source is. So we may be pretty large, but every single cell in our body has to be within, according to Krogh, within at most a few hundred microns of the source of oxygen. Inhaling that oxygen in the lungs, but then the cardiovascular system has to distribute it through all the organs, but even when it gets to the organs, it has to distribute it properly within the organs to get right down such that each individual section of tissue gets enough oxygen. That led to the concepts of capillary recruitment and a number of other ideas that have been generated from that. So here's August Krogh, won the Nobel Prize back in 1920. He published his paper in 1919, and he won the Nobel Prize in 1920. Rather remarkable. He then came over to Canada, met Benjamin Best, who had just won the Nobel Prize, I think, in 21, and took back insulin to Europe, and that's the source of the pharmaceutical industry supplying insulin to the rest of Canada. So just a small little bit of information. So fundamental law of diffusion governs how large a cylinder of tissue. It really governs the density of the capillaries in the tissue and must be supplied precisely where it's needed. So the concept would be each capillary is supplying its own particular cylinder. And that's sort of what it would look like. Now, again, low consumption, large cylinder, high consumption, small cylinder, there must be capillary recruitment. What have I been studying? He's already figured all this out more than, not quite 100 years ago, but it's getting pretty close. Well, we've developed methods for studying oxygen transport in vivo to test some of Krogh's hypothesis. We use a dual wavelength video microscope and principles of light absorption spectroscopy to measure oxygen levels in those videos that I just showed you, so we can measure them. So our setup is simply a microscope, and I've seen some beautiful microscopes here. I wish I actually had access to some of your microscopes. But here's a simple Olympus microscope. It has a high-intensity light source. Here's the rat on the stage. There's a little spot of light there that's being shone down through and objectives underneath. Beam splitter to send the light to 2 separate digital video cameras, and then it's captured live streaming onto the video monitor. And the critical thing is each camera looks at a different wavelength of light. So I'll talk about that a little bit more later. But here's the working area. Here's our rat on the stage, hooked up so that it's giving fluids. We measure blood pressure and keep the animal happy for anesthetized and happy for 5 or 6 hours, depending on the experiment that we're doing. This just gives you, for the engineers in the room, which I usually don't end up talking to engineers in the room, this is just a quick overview of the whole system in terms of what we do. So here's the rat on the stage of the microscope, beam splitter sending it to 2 digital video cameras, store the video sequences, process them. So there would be a single frame, that would be a processed frame. We use all of our work is done in MATLAB just because it's simple to work with. From these images, we can collect geometry information, measure hemodynamics, so blood velocity, essentially how many red cells are going in the vessel, how many are going through per second, and cell spacing. And we can also measure the oxygen saturation with position. And this, of course, is occurring over time. We do this on a frame-by-frame basis for anywhere from 1 minute to 10 or 12 minutes, depending on the study that we're doing. So you collect all this information, and it provides, in the 2 images that we've got, we have everything about geometry, everything about the blood flow, how much oxygen is being carried by the red cells. So we have a full description of oxygen transport. If we then focus down through, or I should say focus up through the tissue, because we're looking at it from below, we can collect geometric information at different planes within the tissue, and we can do it overlapping fields of view so that we can generate 3-dimensional maps based on the red cells flowing through those vessels. With a majority of these vessels will have information on their oxygen transport. That thing goes directly into a mathematical model where we can now start to predict what would be the partial pressure of oxygen within that tissue. What we measure is how much oxygen is being carried by the red cells. The red cell, is it 100% saturated with oxygen, or is it 50% saturated? Depending on its oxygen saturation, that will determine what the partial pressure of oxygen is in the capillaries and how far that oxygen can diffuse out into the surrounding tissue. We can't measure in 3 dimensions what the partial pressure levels would be, but using a mathematical model, we can predict what that would be based on knowing the geometry and the flow. Okay? One of the interesting things is as soon as you do that, you realize that Krogh thought of this capillary right here as totally in isolation, as if it only supplied oxygen to the surrounding tissue that's right around it. It didn't diffuse oxygen to or from other surrounding capillaries. There wasn't diffusional exchange amongst these vessels because he set up a boundary condition in his model, which is 0 flux on the edge of the tissue cylinder. So really, that capillary only supplies that cylinder of tissue without any talking to any of the other vessels that are around it. But when you get vessels that are clustered like this, all of these vessels together are supplying oxygen to the tissue. And if one of them happens to have too low a supply rate of red cells, it will pick up oxygen from its neighbors and continue to distribute that oxygen further. So that's something that comes out of when we do the mathematical modeling. If it would have been Krogh, we would have seen little circular cylinders of colored light here indicating what the partial pressures are. This turns out to be a much more uniform distribution of oxygen, and it's really, we need to think of it as being all of these vessels in that volume are contributing to the oxygenation of that volume, and that's a really important thing. So we're no longer talking about supplying oxygen just down to 100 microns. As long as we can get the oxygen distributed to this network of vessels, they will do the rest to make sure it's distributed. That's in healthy, normal tissue. If we have a disease where we start to lose capillaries, like diabetes, or one of the other diseases I study, then the diffusion distances get too great and they no longer can exchange oxygen between themselves, and then you get into a lot of trouble. Right? Hopefully I can reinforce this as we go along. So we process the videos to get, there's a short clip of red cells going through the vessels. Notice how non-uniform the lighting is across, because there's probably deeper down, there's a larger vessel that's absorbing a lot of light. Over here, there's no large vessel in the background, so it's not absorbing too much, so it's kind of dark. It's kind of bright over there. We process it so that we can actually see where the vessels are that are in focus right at the surface, so that helps in terms of our analysis. Another way we do it gives us a little bit more information on all the vessels that are there. It's another processing technique. And then finally, this one gives us actually a measure of the optical density in every pixel in that image. And it really does a nice job of laying out where those vessels are. So you can see the vasculature. You start to get an idea of how we put together that three-dimensional geometry. This is just the software that we've written where we go through and we select vessels that are going to be analyzed. So to make it quite clear, we don't get data from all those vessels in that field of view. We haven't set it up to be able to do that. We pick out discrete vessels that are clearly in focus, and the software measures the dimensions of those vessels, provides us with light intensity information from the vessel that we can use to calculate all of our hemodynamic data. And here's the absorption spectroscopy, the idea being white light shone through the muscle. It comes through the hemoglobin. The incident light is the light that we would see when the red cell's not there, and then we measure how much light is being absorbed by measuring the light when the red cell is there. So we look at it two wavelengths, calculate an optical density based on the log of the ratios of intensities, and then we've got a linear relationship with saturation and the optical density ratio. We do an in vivo calibration. Here's the extinction coefficient versus wavelength of light. I think some of you are using near-infrared spectroscopy. You're working way out in the red area of the spectrum, so you're way out here and even further, where the amount of light absorbed by a red cell is really quite small. And if we were to look at those wavelengths with our microscope, those red cells would be transparent in those images. You would not be able to, you could only tell the red cell was there by the membrane. We work down on this end of the spectrum, down in the purple. I usually say the blue, but really down in the purple, where the extinction coefficients are really quite high, so that our red cells absorb a lot of light. It means I can only work right at the surface. You use red light so you can penetrate deeply. You use the purple light so that you get high contrast on the cells. And as we change their oxygen saturation from low to high, we change the extinction coefficient over a large range, and we can measure changes in light intensity, measure the optical density quite easily. And from that, we've got good measures of the oxygen saturation. So to get down to look at single red cells in capillaries, then we have to work at that end of the spectrum. You're trying to get an overall global measure, then you work up higher. Is that clear? And this is what our analysis looks like. So you saw we selected some vessels. We get what we call space-time images. They're the light intensity along the vessel, which shows streaks, the streaks being the red cell and plasma as it goes through. And we then calculate velocity, hematocrit, how many red cells are in the vessel, the supply rate, and also their oxygen saturation. Just the way we do our spatial correlation of data. So here is our velocity in frame by frame over, in that case, about a minute's worth of data. This is reconstructing the capillary network. So here is overlapping fields of view. This is Graham Fraser's PhD work. So those are individual frames. It's very difficult in an individual frame to see where the capillary is because we don't see the walls of the vessels. But we can tell where the vessel is by the motion of the red cell as it goes through the vessel. So that's what we're measuring, is essentially the motion of the red cells as they go through. And now you can see quite clearly and distinctly where those capillaries are by detecting where the red cells are actually traveling and changing its intensity. Then if we do it on multiple layers and reconstruct that, we can get a full three-dimensional reconstruction of that capillary network. And it's not like we had taken a fluorescent tag and taken the tissue out and used a confocal microscope to look at it, which would be dead tissue. We are doing it based functionally on which vessels are actually carrying red cell flow. So this network is a functional network telling us which vessels have actually got red cells through them. There might be some other vessels in there which are not being perfused with red cells, and we wouldn't see them. If we change positions, they may pop up, we might see them. Or if we have a disease, we might start losing some of those vessels and we'll be able to see that as well. But with that, we can then put the geometric data into Dan Goldman's mathematical model, and we can start predicting what the partial pressures of oxygen would look like in the tissue around that particular vessel. So it started from the venous end, and it's worked up to the arteriole, and that's going from the arteriolar end down to the venous end of that vessel, of that group of vessels. That's sort of an overview of a lot of the math, of the technology and the modeling that we do. Now let me go on and give you a little more detail about...
Can I just interject a small question for my own benefit?
Sure.
My recollection is that your PO2s, your oxygen transport, is also temperature dependent. We've been mentioning about temperature here, and one might make a case that, in fact, oxygen transport and temperature distribution are co-regular.
Well, temperature, of course, reflects the metabolism in the tissue, right? Level of metabolism. In athletes, when they exercise, their temperature goes up dramatically because of all that energy that they're expending. In our case, we're looking at resting skeletal muscles. We're not exercising it, but we do make sure that it's temperature controlled. We don't...
Can I ask you what temperature?
Well, we don't, we maintain the animal. The whole animal is at 37 degrees centigrade. We keep our muscle at around 35, sometimes somewhere between 32 and 35, but usually around 35 centigrade.
Do you have a tag on blood pressure?
We measure, these are excellent questions because we monitor continuously blood pressure and heart rate in our animals. They're on a ventilator, so we maintain, we measure their blood gases and their oxygen saturations in arterial blood, and we keep them well oxygenated. We keep them up above 95% oxygen saturation in the arterial blood. We also check glucose and lactates and things like that as well.
But do you regulate blood pressure? Do you monitor blood pressure?
Oh, we monitor the blood pressure, and we have them hooked up with fluids so that we can give transfusion. We give them fluid resuscitation on a regular basis to keep the blood pressure up. We use an anesthetic. We use pentobarbital as our anesthetic that we use, and we found that that gives us, you have to worry about temperature regulation and respiration with pentobarbital because the animal is, respiration is suppressed. So that's why we have it on a ventilator. We have, the ventilator is actually on, we don't just give it straight air, we give it a mixture of oxygen and nitrogen so we can adjust that to keep their saturations up. And we give them fluids to keep their pressures up. And we monitor heart rate on continuously. So in terms of being an engineer, we worry more about the physiology, the baseline physiology of the animals we're studying than many of the physiology labs. They make one measurement 10 years ago and say, if we do these conditions, the animals will be quite healthy. They don't worry about it. We're doing a lot of stuff with clinicians, so we really worry about trying to keep things controlled. We also, I didn't really talk about it. Actually, I'll show them the next picture. Our muscle is sitting on top of a glass stage. Glass it's covered with a glass cover slip, and then there's saran wrap, the old-fashioned saran wrap, to keep it isolated from the surroundings, and it's got moisture in there to keep it healthy and moist. Most labs will suffuse a solution over the top of it that keep it moist and has a low oxygen level, etc. But if you do that, you're really altering the conditions of the muscle itself. So we try to allow the muscle to set its own blood flow level and its own oxygen levels by isolating it from the surrounding environment. And then this is one thing we probably should go back and think about. Thank you for reminding me. We should be measuring the temperature of our muscle more frequently than we probably do. We've got a little thermocouple we slip in there, but not all the time do all the people use that. Does that cover all?
For now, yeah, sure.
Okay.
I could chat with you for half a day on this. You have a problem with me, and that is I have a big, major difficulty dissociating oxygen transport from temperature distribution. Temperature regulation, blood pressure regulation, which is your oxygen transport system, are intricately co-regulated, and I have problems dissociating the two.
Well,
you're talking about an oxygen dissociation curve, which is big for blood pressure regulation people. All right, but I'm not sure I want to get into it at this point.
Well, maybe we talk about it another time. Okay. I think that when it comes, I'll show you data that will show that we have O2 regulation independent of blood pressure and independent of temperature. I can show you that in a few minutes. Okay? Here's, if we take baseline data with that system, where we've isolated the tissue entirely from the environment. Baseline, so we're just looking at it over a 3-hour period. Baseline average velocity of our red cells, about 125, 130 microns a second. Roughly, in this case, it's about 9 cells, but I usually think 10 red cells a second go through a capillary. That's a good round number to think about. Hematocrit is, in these animals, was about 17.5%. Systemic hematocrit, if we took a blood sample from an artery or a vein and spun it down, it'd be about 40 to 45%. But in the capillaries, it's down around 17.5%. And that's one of the interesting things that has confused many people for many years. Why are the hematocrits and capillaries quite so low? I'll give you some ideas about that as well. Oxygen saturation in the capillary, this is an average value at arteriole and venular end, is around less than 65%. So entrance into our capillary bed is usually about 65 to 70%. Arterial blood, I just told you, is around 95%. But by the time it gets to these capillaries, we're down to a much lower level of oxygen saturation. It's something that Krogh didn't know anything about in terms of, he thought that the entrance PO2 in a capillary was the same as arterial blood. And that's what you would sort of think, but it's not. And we're not the first to show this. It's been known since the early 70s that there's a drop in oxygen along the way. And you can see our system is fairly stable, at least over these 3 hours that we're looking at. So average capillary hematocrit, it's a volume fraction of red cells in the capillary volume, about 17%. Should be around 35 to 40%. We measured from the diameter of the red cell column in our images, so we're actually dealing with, we shouldn't have an error. Some people measure out to the edge of the capillary, and depending on how you view that, you may not get the right volume. We've just used the red cell column itself, and yet we still get low values for hematocrit. So what's going on? Why is it half of systemic? Well, the same biophysical mechanisms that results in low capillary hematocrit also results in our low oxygen saturations. It's something called the Faraeus effect. It means that red cells tend to migrate away from the wall of the vessel, leaving a layer of plasma. And so red cells go through the vessels faster than plasma does. Red cells have, if you measure transit time of red cells versus plasma, red cells get through faster. In if you do the mass balance on it, you realize that you'll end up with, under those circumstances, a lower number of capillaries in your tube, a lower number of red cells in your tube when the red cells are going through faster than the plasma. So your beaker that you're collecting the blood in at the end here, your discharge, will have still systemic hematocrit of 35%, but the red cells are going through the tube faster than the plasma. And take a snapshot of that capillary tube, you'll end up with something like maybe something lower than systemic hematocrit. That's part of it. And then there's also a flux of oxygen out of the arterioles into the surrounding tissue, so we have lower oxygen saturations near the wall. So I've got a couple of cartoons for you. Here is the Freitas effect. Here's the red cells going through a vessel in an arteriole. There's lower hematocrit around the edges and a higher hematocrit in the middle. And as we separate out these red cells, we get a slightly higher hematocrit in the main vessel and a lower hematocrit in the smaller vessel, because we've siphoned off the red cells that are close to the edge, where the hematocrit is lower. So, get that? That sort of makes sense? We go on, they've got lower saturation. So these are the ones with lower saturation. Let me back that up. The lower saturation ones go down this side, resulting in low saturation in those vessels, and the higher saturation stays up here. If we now dilate the vessel, okay, back that up again. If we dilate it, we now start to sample further into the flow stream, and we get a higher hematocrit and a higher set of red cells with higher saturations. If I vasoconstrict, I'll get even a lower hematocrit and a lower oxygen saturation. So it's a nice cartoon. Does it actually occur? I'll show you some data that would support that. So it means that, what does this mean? If I increase my oxygen demand in this area of the tissue, and I vasodilate a vessel to send blood flow over in that direction, as soon as I vasodilate that vessel to send blood flow over there, I'll increase not just the velocity of the red cells, but I'll increase the number of red cells, I'll increase the hematocrit, and I'll increase the saturation. So the impact of vasodilation is an immediate amplification of oxygen supply in that area. And if I vasoconstrict, I'll also have the same type of problem. If I did something that erased the radial gradient in hematocrit and oxygen saturation, like exercising heavily, where I'm really sending a lot of blood flow down to my organs, I wipe out those radial gradients in saturation and hematocrit, the impact will be that I'll have a uniform distribution of blood flow and oxygen at the bifurcations, and again, an immediate impact on oxygen supply and amplification. So rather than recruitment of individual vessels, which is what Krogh was talking about, we talk about a recruitment of red blood cells with higher oxygen saturation to generate a more uniform distribution across the capillary bed. Not capillary recruitment, flow recruitment. And it's flow recruitment with higher saturation, etc. So this just shows bifurcation and variability in hematocrit coming off of it. I've added something in now that actually I think shows this a little bit better than that particular one. So here's a single capillary that we're looking at here. One of our space-time images, so position along the vessel, looking at the red cells as they go through over time. This is for about, I think this was with a different camera, so it's a different frame rate, so that's still about a minute's worth of data. If we take a look, when the red cells are almost zero velocity, they're fairly stationary in the capillary. As they increase their velocity, they become almost vertical streaks. And here they slow down again, in fact, reverse slightly. So we see that variability in velocities, but we also see there's quite a variability in how many cells are there. So here's the plot of velocity going through different peaks. It starts at 0, peaks up to about 200 microns, comes down to 0 again, goes up to about 250, almost 300 microns a second. So in this 1 minute's worth of data, we have very large variations in the velocities of the red cells. Large variation in the hematocrit or lineal density as well, from almost down to 0 for a moment and much higher. Product of these two gives us supply rate, the number of cells per second going through. And when it's stopped, as we stationary here, we see there's no flow of red cells, very, very little flow of red cells going through that. The color coding is saturation. When we've got low velocity, we end up with low hematocrit. And we end up with very low oxygen saturations by looking at the color coding here. I'll show that again in a moment. When we increase the velocity, so a vessel is vasodilated, we increase the concentration of red cells in the capillary, we increase the supply rate. So over this period of time, we've had large variability in hematocrit, supply rate, and saturation, all because of what's going on at that bifurcation just upstream. As it vasodilates and vasoconstricts, we're sending through red cells of different saturations, we're sending through blood of different levels of hematocrit. And here's what our saturations would look like over that period of time as well. Spiking up to almost 100% saturation and getting down in some cases to about 40%. So that's the system that we're working with. Now we want to start studying regulation. Boy, I better keep moving fast here, I'm running out of time. So here's the system we use. We put a well, I call it microfluidic. After I see your guys' microfluidics, I wonder, ours is really crude compared to what you do. But we create a chamber here which is transparent and very thin, allows us to put gas through at variable oxygen levels. We have an oxygen permeable membrane right under the muscle, so we can add oxygen into our muscle. Partial pressure average is around 40. If we put 55 millimeters of mercury, slightly above normal, we can drive oxygen into the muscle. If we want to take oxygen out of the muscle, then we go to 15 millimeters of mercury, lower, we suck oxygen out of that muscle. So we can vary it. These are over nice physiological ranges. This is not like super high levels of oxygen. We're not going down to 0 where we might have some effect on metabolism as well. And this is what a video looks like. We're starting at 55. Right at the beginning, I've just switched it to 15. And now watch the blood flow. There's about a 10-second delay. And here is the red cells now flooding in to that to compensate for the fact that we've got low oxygen. Now, this is all temperature controlled. There's no change in temperature. There's no change in metabolism. This is just simply changing oxygen. Here it is processed in a way that you can see it a little more clearly. Here it is with high level of oxygen, high, 55. And then we switched to 15 a few seconds ago, and now you see the influx of red cells into this tissue. So the gas is temperature controlled, muscle's temperature controlled. All we've changed is oxygen levels. Kept CO2 levels the same, it's just oxygen. You can see what the response is. If we measure it, you can see that high levels of oxygen, we have a PO2 sensor in the outflow. We can see these low hematocrit when we have high oxygen, high hematocrit when we've got low oxygen, and it almost looks like it's being regulated back down as the hematocrits are coming down before we give high levels of oxygen again. And we look, here's the red cell saturation. Here, when the new cells came in, they came in at a low saturation, but quickly spiked to a high saturation, higher than what we normally see, and here they are being regulated back down to about 65%. So you're actually watching the regulation going on as it's trying to regulate that. So that would be a typical saturation. If we put a sine oscillation in, I think sine oscillations, sinusoids, we can get it to actually follow the sinusoids beautifully in terms of saturation and in terms of supply rate as well. How does it do this? And I'm almost out of time, but I'll zip through quickly. How does it do it? This is what the arteriolar tree. These thin dark lines are arterioles, and this is a scale of 1 millimeter. How does it distribute the oxygen and the red cells appropriately in that system? Because we're coming from up here, I'm really stimulating a little area that's just tiny down here. How in the world is it able to adjust that? Okay? Well, some people said it was because we had a direct effect on the arterioles. We're changing oxygen levels over the arterioles, and so the arterioles sense that, and they respond. Well, we thought, this is a different muscle than the one we use. I use it for these purposes because you can see the vasculature so clearly. Our big vessels like this are buried deep in the tissue, so they're not anywhere near where that oxygen changes are. So I was pretty sure we were not affecting our arterioles. Skip through this. So every bifurcation, you've got to distribute the red cells correctly at every bifurcation to make sure you got the right amount in the right place. A venule and then taking it all the way back. There's our 10 power field of view in this image. Here's our 20 power field of view, just to give you the scale. Right? Here is what, if we go down to that scale and we look at an overlapping series of images, here's a little arteriole here, supplying capillaries in this direction, a little venule there, from this arteriole down to this venule here. It's a really complex network. It's not nicely ordered, like I see with your beautiful microfluidics, where everything you've made is lined up gorgeously. This is what nature is dealing with, right? How does it regulate the right amount of oxygen to this arteriole? And because this one over here may need a different amount of oxygen, how does it regulate it within that particular situation? And how do we test that in vivo to look at it? Whoa. I hope I show that later. We put a little window right here. I hope I've got it in the slides, where we change the oxygen level just over capillaries. So that's where our microfluidics comes in. We look at just the capillaries. Okay. Need to be able to sense the oxygen in the arteriolar and capillary bed. Need to be able to send a signal upstream to the arterioles, because they're the ones that turn the taps on and off. And what we've been working with is the idea that the red blood cell, what carries the oxygen, is also the sensor for how much oxygen it's got. It knows what its oxygen saturation is, and it signals the vessel wall to tell the vessel, I've got 75% full of oxygen at this moment. And it sends out a signaling molecule, ATP, to the vessel wall. The receptors on the vessel wall conduct a signal from the capillaries through the endothelium, the cells that line that capillary, all the way back up to the arteriole. So it's, how many of you are electrical engineers? This is all electrical signaling within the microvascular bed. It's ion flow, but it's electrical signal that's being sent from the capillaries to the arterioles, and the integrated signal from all those capillaries tell the arterioles how dilated they're supposed to be. At least that's our hypothesis. And we think it's the red cell, and the change in saturation of the red cell that results in the release of ATP and causes that conductive signal that's going along. Those endothelial cells are about 100 microns in length. They're electrically coupled with gap junctions, so if you disrupt that, then the signal should go away as well. So as oxygen is being released from the capillaries, and from arterioles into capillaries, and from capillaries into venules, it's readily to diffuse everywhere. We're also, whoops, we're also releasing ATP into the surrounding tissue as well. And the amount of ATP that's released will determine the level of hyperpolarization and the signaling will occur. And so we have all these individual units of capillaries down here sending a signal back up to the arteriole. These are all integrated as they go along, just like they would be with electric signal, and determining what the size of the upstream arteriole should be for sending the appropriate level of flow. And if one increases demand, it'll dilate more and send more flow in that one particular direction. You have to integrate this response, because if this vessel down here said, I need twice as much flow, and you don't dilate the ones up above it, where does it get that extra flow from? It steals it from its neighbors, right? So if I double the flow into here without having a corresponding increase in flow that corresponds to that, I have to steal it from other vessels. So the whole system has to work as an integrated unit. And the way we tested that is using a micro window over our capillaries and just changing the oxygen levels just here. So here's our chamber, but now it's with a little tiny outlet for changing oxygen levels. Here's our crude micro outlets that we were using. The one we ended up with was 100 by 200 microns. But we tested others, did mathematical modeling to make sure that what the effect would be on the capillary bed. We can talk about that later, but this is the key stuff. Here's the window here. Here's overlapping fields of view showing the vessels. Here's a vessel just outside our window. And here's one in the window. Here's our functional images where we've overlapped them so you can see the vessels in the window and the ones just outside. We're going to do analysis on oxygen levels here and out here as we oscillate the oxygen in the tissue, or in the chamber, in the window. The first set of data is coming from these 3 segments that we analyzed. So this one's up close to the arteriole. This one is a little further, this section is further down, and this is just beyond the window. Modeling would say we shouldn't have much of an effect on the oxygen levels in this capillary because it's closer to the arteriole. As we go down, the level of oxygen that we supply should be increasing. So the changes that we see should be larger in these segments. And that's what we see. This is the top segment of the capillary, very small changes in saturation as we alter the PO2 in the chamber. As we alter the PO2 in the chamber and look further down, we see larger and larger changes. And we also see the blue symbols indicate there's a change in supply rate because we were affecting the supplying arteriole. Here it is for the vessel that was in the window. We changed the oxygen level and we increased the saturation. And we reduce the saturation, and we go back up again, and we see corresponding is oxygen levels are high, saturation, the supply rate goes down, oxygen levels are low, supply rate goes up. Repeated 20 minutes later, we get the same response. We look at it for 10 capillaries, average all the data, put it together, we see the same type of response as oxygen levels in the chamber go up. The supply rate reduces. As we reduce the oxygen in the chamber, the supply rate goes up to deliver more oxygen. So we're showing flow responses in capillaries in response to just changing oxygen levels just in the capillary itself. So whether it's the red cells or not, we can't say yet because we didn't test ATP released red cells in these images, but we do show that the signal is coming from the capillary bed up to the arteriole to alter what the flow rate will be. And it's altering it in the appropriate direction. So, microvasculature is able to sense its low oxygen environment. It is able to signal to the arteriole and create. Capillaries can essentially control their own distribution of oxygen supply. Future work, we need to demonstrate it's the erythrocyte that's actually responsible for this and verify that it's actually conduction. Others have we've done individual parts of this, but we've never put it all together in an in vivo study. We've really just started by demonstrating that capillaries can signal. Going back to Krogh, it's not capillary recruitment, as Krogh had said. It's actually regulation of oxygen supply by regulating those terminal arterioles to deliver the right amount of oxygen to that unit of capillaries. And that unit of capillaries then makes sure that the oxygen is distributed to all the tissue that's needed. Sorry for just pounding that out at you so fast. I'm having so much fun doing this type of research that I have to tell you the whole story. And I have to throw in other bits along the way with it as well. So it's hard to get me to tell you less. Any questions? Yes?
Yeah, so that was a really interesting talk, and coming at this from a neuroscience perspective, I'm more familiar with the neurovascular coupling mechanisms that have been studied and previously knew nothing about muscle. But what I was wondering is a lot of the neurovascular coupling literature is started to really implicate lactate and other metabolic couplers. Basically, as neuronal activity goes up, there's increased local metabolism, oxygen drops down, and through glycolysis and other processes, lactate goes up. And one of the main ones that Brian McVicker at UBC and Grant Gordon at U of C have shown is that lactate is really, really important in the neurovascular coupling in terms of regulating the supply and demand locally. Has anybody looked at lactate in muscle?
I don't know if they've looked at lactate in muscle the same way that they've looked at lactate in brain. We're not altering lactate levels when we're doing this.
Oh no, sorry, that's all like from the normal in vivo processes, the normal metabolism.
So
it's endogenous.
Yeah. So when they've looked at lactate in the brain, how have they done that?
Well, they've done it with a bunch of sophisticated calcium uncaging things and fluorescence and things like that. Actually, there's a few groups here that I'm sort of going to be working with, trying to look at this with metabolite-specific biosensors, essentially, which are little like microelectrodes. We've just bought a biosensor rig to look at these lactate changes.
So I'm sure that lactate plays a role in regulation. The body doesn't just focus on one single regulatory system, right? And even I point out here that we think it's the red cell, we think it's the red cell signaling within the capillary. We've tried to do it in a way that we're not altering metabolism, so that lactate production shouldn't be changing. But I can almost guarantee that there's not just one control system that's associated with regulating oxygen delivery. There'll be multiple at different levels within this vascular tree. I'm looking at only one aspect of it. I would imagine that lactate levels would also result in, if we increase lactate, we'd probably see an increase in blood flow. There's probably changes with carbon dioxide as well. And all of these things will play some role. Just changing metabolism on its own should have an effect.
Yeah, and that's sort of what I was wondering is like as you decrease the oxygen availability within the tissue by lowering the oxygen, if that might inadvertently change some of those metabolic processes by invoking more glycolysis and so forth. So
one of the reasons we don't go below 15 millimeters of mercury in a chamber is to keep our oxygen levels high enough in the tissue that we're not altering metabolism.
Okay.
So that was a conscious decision. The very first experiments we did, of course you put the oxygen down to zero because that's going to give you the biggest response. And then we stopped to think about that. Well, if we put it to zero, it's like in the chamber, it's likely not zero with the tissue, but it's going to be low enough that someone's going to question us and say, we think that you're affecting metabolism. Most people would say 15 millimeters of mercury is enough to maintain aerobic metabolism in our tissue. We're not actually altering lactate levels or mitochondrial function at that level. And so then the other question is, when we did it with this little window, we're really only affecting a very small part of the muscle fiber. If it were a change in metabolism in that area of that muscle fiber, how did that muscle fiber get its signal to the appropriate arteriole to change blood flow? It's okay if you think about it globally, if we increase metabolism and lactate levels go up, and it's uniformly going up throughout the tissue, then the signal would be right there at the level of the arteriole to cause the vasodilation. So you have to have something that's going to have a conducted signal that goes from the capillary up to the arteriole. Now I know in the brain, in neurovasculature, they also talk about astrocytes, actually the feet of the astrocyte coming in on the capillary and altering the diameter of the capillary. Well, that will have an impact up to some level because it will alter the flow in individual capillaries, but it's not going to result in the increase in arteriolar flow that's actually going to substantially be able to increase oxygen delivery into that area. So I think, and a lot of that work has been done on brain slices, right? that it has intact tissue, but it's not got an intact network of vasculature, and it certainly doesn't have blood flow going through. So I think there's a whole area that's just still massively wide open for studying regulation in that environment. And if I get a CFI grant, which I'm looking at right now, I may have some microscopes that I can actually start looking at the brain as well. So if anybody ever reviews the CFI grant that's going in from Western, please fund it, okay? You start asking questions.
Yeah, I'm partly confused, I must say, and I don't know whether I have it right. And so you measure oxygen saturation of hemoglobin because of the capillary system.
That's correct.
And but you're interested in oxygen delivery to the tissue, i.e. to the mitochondria where it's exact.
Yeah.
And my gut feeling is that the rate limiting step in this process is when the oxygen comes off the hemoglobin, it has to diffuse a little bit through the tissue and reach the mitochondria and the cytoplasm to use that. I think that is the slow process. And the rate of that will be determined how steep the diffusion rate is. Is that right?
That's correct.
So I read long ago, decades ago, that for instance, myoglobin muscle plates are and it binds as hemoglobin, and then it may actually also
Facilitate diffusion. Yeah.
What do you think of that?
So the muscle that we use, I should have mentioned, is the extensor digitorum longus muscle. It doesn't have a lot of myoglobin. So we're sort of lucky in terms of that. But the other thing, I think if you get to low enough levels of oxygen, myoglobin probably does play a role. But myoglobin is fully saturated at about 10 millimeters of mercury, partial pressure. So we are working, in the only facilitated aspect of myoglobin would be shuttling of oxygen from a saturated myoglobin to a desaturated myoglobin, or partially saturated. That's where you get that shuttling effect going through. So if we keep our myoglobin fully saturated with oxygen, then it's not playing a role in what we're taking a look at right now.
But my gut feeling is that oxygen diffusion through your liquids or tissue as a gas is a slow process. It's frustratingly slow. And I think, but I'm not sure of that, when it's bound, the hemoglobin, it doesn't move because of the red cell, but it's caged in. But if it's bound to myoglobin, the myoglobin molecule, which are without oxygen, I think diffuses faster than oxygen atom to fall.
Well,
am I right on that?
No, myoglobin is a much bigger molecule. Oxygen, diffusion of oxygen will be much faster than diffusion of myoglobin. Because it's a gaseous molecule that can move through the tissue faster than that large myoglobin molecule, which has got a heme group associated with it. So it doesn't transport as quickly. The only way that myoglobin, at least my understanding, the only way myoglobin facilitates oxygen transport is if you've got a myoglobin molecule here that's fully saturated. You have another one over here that's not fully saturated. Actually, it's not saturated. It'll shuttle oxygen to that one. And so that facilitates the movement of oxygen from one myoglobin to the next. It's not the actual movement of the myoglobin in the tissue.
Maybe I was wondering about this comes from having worked decades ago with mitochondria, and whenever I can probably equilibrated by a medium which has saturated with oxygen, or help produce. My experience was just that it sits there, the diffusion of oxygen is a very extremely slow process.
So,
but I could be wrong, but maybe myoglobin diffusion is even slower.
The challenge of the microvasculature is, you're absolutely right. Compared to transport by hemoglobin in red cells, hemoglobin in red cells, that transport is very fast. It's carrying a lot of oxygen. But really, the challenging part is the diffusion from the red cell to the mitochondria. That's why we have to have regulation very close by, right? And with Krogh, he thought it had to be really close by each individual capillary, because it was just, and that's what the modeling showed. What we're saying is, as long as the capillaries are close enough together, they'll work together